Hello, we're glad you've joined us for this live webinar, Glycosylation and Stabilization of Programmed Death Ligand 1 Suppresses T-Cell Activity. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Essen Bioscience. Essen Bioscience, a Sartorius company, develops and manufactures instruments, software, reagents, and consumables that help researchers to remotely visualize and quantitate many cellular processes over time within the incubator environment. The IncuSite system is a real-time quantitative live cell analysis platform that enables visualization and quantification of cell behavior by automatically gathering and analyzing images around the clock within a standard laboratory incubator. The system allows researchers to make time-lapsed, fully kinetic measurements from living cells over days and weeks, providing insight into active biological processes in real time. To learn more, visit www.essenbioscience.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Chia-Wei Li, Ph.D. and Instructor in the Department of Molecular and Cellular Oncology at the University of Texas MD Anderson Cancer Center. In 2003, Dr. Li entered the Graduate School of Biomedical Sciences at the University of Medicine and Dentistry of New Jersey. Following graduation, he began his postdoctoral training with Dr. Mien-Chie Hung at the University of Texas MD Anderson Cancer Center, with a special interest in studying the underlying mechanisms of breast cancer metastasis. Since he became an instructor, he shifted his research interest to study breast cancer immunosuppression in hope of understanding of the molecular interaction between cancer and infiltrated T cells in the primary and metastatic tumor microenvironment. Dr. Li's research interests lie in discovering the regulatory mechanisms of cancer metastasis and immune resistance in preclinical settings. On the basis of the newly identified signaling pathways, he investigated the therapeutic potentials of natural food compounds, small molecular inhibitors, and monoclonal antibodies to reduce cancer progression in hope of translating these findings from basic research into clinical uses. Dr. Li will now begin his presentation.
Hi, everyone. It is my great honor today to join today's LabRoots webinar to share some of our recent studies on the PD-L1 glycosylation and the cancer immunotherapy. Hopefully, through this presentation, we will have an idea how we translate basic research to the clinical use in a hope of benefit patient at the bedside. Cancer immunotherapy has drastically reshaped the landscape of cancer treatment since the development of anti-CTLA-4 antibody in treating myeloma in 2010. Today, there are over 500 clinical trials, total of 9 types of monoclonal antibody developed by a pharmaceutical company are currently tested at the clinical site in treating 20 types of cancer on over 20,000 patients. So today, our objective is to first understand the new challenge of breast cancer therapy. Our lab has tremendous interest in studying this type of disease for 2 decades. We have also like to take this opportunity to advocate that there is a special need for the breast cancer patient to be breast cancer research to be continually supported. Because the breast cancer mortality it wasn't like 20 or 30 years ago, breast cancer was so devastating. Since the development of hormone therapy or target therapy, the mortality of breast cancer has significantly reduced. So now the 5-year survival rate is over 90%. So indicate that in past 10, 20 years, scientists do achieve a great advance on treating this type of disease. However, this disease still have a lot of issues, such as recurrent metastasis or drug resistance. So therefore, we still would like to take this opportunity to continuously advocate for the patient that there is a tremendous need for federal government, for philanthropists, for basic research to focus on this type of disease. And second, we would like to introduce what we have been finding in the past 3 years. We focus on the glycosylation of PD-L1, and we translated this study to immunotherapy strategy development. So we would also touch base on that to tell you how we identify founders of this important regulatory mechanism. And lastly, I would like to briefly introduce some of the new technology we developed in the past 3 years. By working with the SM Bioscience, we developed several state-of-art type of methodology to try to help us to understand about basic of science and try to translate this research to clinical use. First, breast cancer, as I just mentioned, has been tremendous treated in the past 20 years. So, however, American Cancer Society still estimate that in the 2016, the new case identified in the woman, there will be 29% of the patient will be identified as breast cancer. So this suggests that although the mortality has dramatically reduced for this disease, however, the incidence of this disease is still very high. So therefore, we as a basic scientist, we still have a lot of room to try to improve the treatment or the quality of the breast cancer patients. One type of breast cancer of our interest is called triple negative breast cancer. This subtype of breast cancer lacks of estrogen receptor, progesterone receptor, and the human epidermal growth factor receptor 2. Because this type of disease lacks of the treatment targets such as above mentioned the 3 molecules. So therefore, there is basically no treatment for this type of disease. Patient diagnosed with triple negative usually goes through the traditional chemotherapy. Unfortunately, only half of the patient can benefit from this therapy, which means another half of the triple negative breast cancer has basically no drug to cure. So to our interesting, we found out that among all breast cancer subtypes, triple negative breast cancer has the most abundant tumor-infiltrated lymphocyte. This lymphocyte present in the tumor supposed to help us to against the cancer growth. But somehow this triple negative breast cancer has the mechanism to silence, to inactivate this lymphocyte inside the tumor area. So this also gives us a hint that in the triple negative breast cancer, if we use the immunotherapy to reactivate the tumor site lymphocyte activity, will be a new therapeutic strategy to enhance the triple negative therapy. Today, one of the most important immunosuppression mechanisms has been identified. Is called PD-L1 and the PD-1 signaling axis. So the next slide showing you is a working mechanism of this molecule. So as you can see on the panel A, the cartoon shows that the tumor cell has expressed a lot of tumor antigen. It's exposed by the NHC1 class on the cell surface. And this presentation, once the T cell recognize this presentation, T cell will come into the tumor area and try to eliminate this cancer cell rapidly. However, cancer cell is also very smart. They will express the PD-L1 on the cell surface. This expression will engage with the T cell on the PD-1, and this engagement will eventually shut off the T cell activity, cause immunoescape. Scientists now have developed monoclonal antibody that can specifically recognize either PD-L1 on the cancer cell or PD-1 on the T cell. This type of strategy will help to mask the interaction surface of these two molecules and therefore block this PD-L1 and the PD-1 engagement in an effort to reactivate the T cell activity, allowing T cell to recognize the cancer cell and then eliminate this foreign substance. Today, this type of immunotherapy has reached a tremendous improvement, focused mostly on the melanoma, Hodgkin's lymphoma, lung cancer, kidney cancer, and bladder cancer. And there are 4 drugs that have been approved by the FDA currently is available, and there are another 5 drugs that is under clinical trial right now, all targeting on the PD-L1 and the PD-1 signaling axis. Then the overall response rate of this type of treatment is around 20 to 40%. Common side effects, including diarrhea, joint pain, autoimmune disease, have been found, but those are mild and can be managed by steroid. And right now, there are more and more questions such as intrinsic and acquired resistance, hyperprogressive disease, and try to identify new biomarkers to predict the response of the anti-PD-L1 and PD-1 is a new challenge that await us to study. So as I mentioned earlier, triple negative inside the triple negative breast cancer, there is a lot of infiltrated T cell. And however, cancer cells still grow. So therefore, we suspect that triple negative breast cancer must have some kind of immunosuppression mechanism. So one molecule of interest is PD-L1. This protein is expressed on the cancer cell, and it's supposed to have molecular weight 33 kilodalton. So as a cancer biologist, what we first like to do is to monitor, to test where does the PD-L1 express in the cancer cell line. So in the panel A, we can see that across breast cancer, melanoma, lung cancer, and colon cancer cell lysate, we feed a simple Western blot. We try to recapitulate the PD-L1 expression on the Western blot. And we found that this protein expression at the molecular weight 33 is very little. So we have tried several ways to enrich this expression, but it was not very successful. So in that time, we actually, interestingly, we noticed above the molecular weight 50 kilodalton, there is a huge abundant without non-specific expression of the PD-L1 because the molecular weight is totally wrong. And it expressed as a heterogeneous expression. It's not a distinct pattern that we used to expect on the regular Western blot. So we didn't pay much attention in that time till we use the overexpression system. With this way, this experiment we did is we cloned 33 kilodalton PD-L1, and we expressed this protein in the 293 cells. We found out, again, there is no 33 kilodalton protein expression on the Western blot. Instead, there is a 50 kilodalton molecular weight of PD-L1 expression. We made several contrasts, and they all seem to act similarly. So therefore, we start to suspect whether this 50 kilodalton molecular weight of the protein of the signal is actually the PD-L1. To confirm this result, we did two shRNA knockdown clone, knock down the endogenous PD-L1, and we found out the 50 molecular weight kilodalton signal was disappeared. When we reconstitute the wild type PD-L1 into those knockdown clone, we found out the molecular weight 50 kilodalton signal was reappeared. We also create another two CRISPR-Cas9 knockout of PD-L1, and we consistently found out that the molecular weight 50 kilodalton is disappeared. So this result suggests that this high molecular weight of the protein is real PD-L1 signal. Again, like I earlier mentioned, our lab has tremendous interest in studying triple-negative breast cancer. So what does this PD-L1 express in the breast cancer? In this slide, we also use the panel of breast cancer cell line, and we group them into non-triple negative and the triple negative breast cancer cell line. And we did a simple Western blot with EGFR and the PD-L1. EGFR is highly expressed in the triple negative breast cancer, which has been known. And the PD-L1 expression also show very high abundant expression at the molecular weight 50, whereas the molecular weight 33 kilodalton here in the red arrow, we don't see much expression. Then we look for the literature to see what might be the reason cause the molecular weight shift of the PD-L1. So one thing comes to our mind is that the protein glycosylation might cause the cell protein molecular weight migration dramatically. So protein glycosylation is attachment of sugar moiety on the protein to form the glycoprotein. It usually occurs approximately predominantly at the N-XT motif. N stands for asparagine, X is any amino acid, and the T is threonine. Glycosylation usually happens in the ER and the Golgi apparatus, and it is estimated that approximately 1/3 of all human protein has this type of modification. They are either N-link, O-link, or C-link glycosylation. Glycosylation controls protein folding, localization, interaction, and enzyme activity. So next, we would like to know whether what we observed, the molecular weight 50 of the PD-L1 is really through glycosylation. So in this experiment we did we overexpress different tags of the PD-L1. In this case, we have MYC tag at C-terminal, we have flat tag at C-terminal, and we create another HA tag at N-terminal. We overexpressing this construct and then treat the cell with tunicamycin. Tunicamycin is a type of chemical that can block N-linked glycosylation synthesis. So we found out when we increase the concentration of tunicamycin, the molecular weight 50 of the PD-L1 start to decrease to molecular weight 33 kilodalton. So this result suggests the PD-L1 is a highly glycosylated protein, regarding as long as we use an overexpression system and or when we look at the endogenous level. So next, we would like to know where does this glycosylation happen. So next, we do the we check the mouse PD-L1 and the human PD-L1 sequence. We found out there are 4 NXT motifs consensusly found out in the extracellular domain of the PD-L1. They are N35, N192, N200, and the N219. So therefore, to understand which site involved in this glycosylation event, we first create single mutation on the PD-L1. And we consistently see that all 4 single mutations has ability to reduce the protein migration of the PD-L1. So then we continuously create double mutant and a triple mutant. PD-L1 migration was continuously reduced till we create a 4-1Q. When we complete mutate 4 glycosylation sites, we found out PD-L1 expression goes to molecular weight 33 kilodalton. And as I showed you earlier, that wild-type PD-L1 treated with tunicamycin also can reduce to non-glycosylation form. So in this case, we can see a nice side-by-side together with these 2 molecules, which suggests that PD-L1 is only glycosylated at these 4 sites. We also create another 3 asparagine mutant at a non-NXT motif, and they do not change any molecular weight shift of the PD-L1. So on the left side, we did a mass spectrum analysis, collaborate with the academic Sinica in Taiwan. We identified this N35, the glycan structure of this N35 PD-L1. Of course, we also identified the other glycan structure, and because of the space issue, we are not going to show you here today. Since PD-L1 is highly glycosylated, so the next we would like to know is whether this fork glycan structure will affect its interaction with PD-1. To do this, we first set out an in vitro binding system. In this case, we overexpression flag tag PD-L1 in the cell, and we use a flag bead to pull down this flag tag PD-L1, and then incubate with PD-1 protein in the solution. And after that, we did a pull down experiment. We found out that the wild type PD-L1 can interact with PD-1 efficiently. However, when non-glycosylated PD-L1 do not interact with PD-1. To test this hypothesis in vivo, we use a InQSight lifetime imaging system to quantify the dynamic change between PD-L1 and the PD-1. So in the first picture we show in the red color is BT549 cells has nuclear IP expression. And then this cell will also express a wild-type PD-L1, which is a glycosylated form of the PD-L1. And then we add a fluorescence-labeled PD-1 protein in the solution. After 12 hours of time, we can measure the dynamic interaction between PD-L1 and the PD-1. So as you can see that there is a huge accumulation of red color on the cell surface after 12 hours of treatment. And on the other hand, in the non-glycosylated PD-L1 stable clone, we don't see any much green fluorescence, suggests PD-L1 and PD-1 do not interact after 12-hour period. So here we show you 3 individual clones. They all act similarly, which consistently support with our in vitro data show that this glycan structure is required for protein-protein interaction. So to understand the function of this the glycosylation, we did a we did a T-cell killing assay. So in this experiment, again, we use an InQSite machine to mimic the dynamic interaction, the cell growth of the cancer cell and in the presence of T-cell. So in this case, it's a BT549 nuclear IP clone, and we add a activated T-cell, incubate together for about 5 days of time, and we can see the green fluorescence, which indicate the caspase-3 activity. As soon as the cancer cells start to die, we will see the reduce of the red color and also the gain of the green fluorescence. And this quantification tells us that indeed glycosylated PD-L1 cancer cell has the ability to resist to T-cell killing, whereas non-glycosylated is very sensitive to T-cell killing. Next, we performed the in vivo experiment to confirm this result by inoculate 41 PD-L1 wild type and the PD-L1 non-glycosylated PD-L1. And then into the biopsy mouse, we found out at 2 weeks of time, there is a huge geometrically gross differences between these two clones, suggests that loss of the glycosylation, the cancer cell loss of its ability to suppress T-cell activity. And we also did a functional analysis, try to understand what's the difference between glycosylated form and the non-glycosylated PD-L1. So in this case, we do the cyclophosphamide post-stress experiment. We found that the wild type glycosylated PD-L1 is very stable, whereas non-glycosylated PD-L1, the half-life is very short. And the two systems all indicate the same phenomenon. So next, we would like to know what's the upstream signaling that regulates this glycosylation event. So what we did is we took 2 triple-negative breast cancer cells, BT549 and 468 cells, and we treat with the different stimulators such as EGF, IGF, HGF, FGF, and the TGF-beta. We found consistently EGF can induce PD-L1 glycosylation in 2 triple-negative breast cancer. Because EGF is a ligand of EGFR, so we also confirm this result by adding different type of EGFR ligand. And we found that as long as EGFR being activated, the PD-L1 expression also increased. And the right panel shows you the flow cytometry measure the membrane-bound PD-L1 is being upregulated by EGF ligand. So lastly, we would like to use this model to conclude our finding that EGFR has the ability to stabilize PD-L1 because non-glycosylated PD-L1 will be recognized by the GSK3 beta and thereby recruit the beta-TrCP to degrade this PD-L1 protein. Once PD-L1 get glycosylated, it will prevent the GSK3 beta recognize the PD-L1 and therefore cause the immunosuppressor activity. And lastly, since our signaling analysis indicate that EGFR is one of the key signaling that induce PD-L1 glycosylation, we suspect that the EGFR, if we use a calcium kinase inhibitor to inhibit EGFR, will be able to downregulate PD-L1 expression in the cancer cells. So here is the flow chart that we perform this animal analysis by treating every day daily with a Genefovir, and also every 3 days we treat with 100 micrograms of anti-PD-1 antibody. And we found out the combination of these two therapies significantly reduce the tumor growth of the 41 mouse cells. And this mouse has increased the mouse survival about 20% by combining these two therapies. We also look at the TIL analysis inside the tumor area, which is CD3, CD8 positive. We look at the interferon gamma expression, and consistently combining these two therapies enhance dramatically interferon gamma secretion. And also we look at the granzyme activity, found out that the tumor area that when we combine these two treatments, the granzyme activity of the cancer cells can be activated. So conclusion of this, the first part of my talk is that we found PD-L1 is unlinked glycosylated at 4-NXT motif. GSK3 beta can phosphorylate and destabilize non-glycosylated PD-L1. And glycosylation can stabilize PD-L1 through prevent GSK3 beta binding to non-glycosylated PD-L1. And the upstream signaling is the EGF that can increase the non-glycosylated form of PD-L1. So if we combine you inhibit the EGFR signaling, we are able to sensitize cancer triple-negative breast cancer cell to anti-PD-1 therapy in the syngeneic animal model. Next, I would like to quickly switch gear to show you one of our another finding. We also incorporate a lot of methodology using the SM Bioscience Intusight microscope, and we have published another paper in the Cancer Cell 2016. So what we found is that triple negative will also induce chronic inflammation. So we would like to know what's the inflammation, why this inflammation can cause cancer progression. So we found that inside the tumor microenvironment, macrophage has the ability to secrete the TNF-alpha, and this TNF-alpha will recognize cancer cell TNF-alpha receptor. So in this enlarged cartoon on the right side, the underlying mechanism is that the TNF-alpha will activate NF-kappa B and cause the P65 nuclear translocation, and thereby turn on the CSN5 expression. And we identified that CSN5 is a novel deubiquitination enzyme, and this enzyme will stabilize PD-L1 and therefore translocate, induce the cancer cell immunosuppression function. So in the end, we found in this study that in the tumor microenvironment, the 2 immune cell, macrophage and the T cell, and how these 3 cells interact together in the tumor microenvironment related to the immunosuppression activity. Next, I would like to spend some time to also discuss our recent study that we work with the ST-Cube Pharmaceuticals. We try to identify the first anti-glycosylated antibody. As we showed you earlier, that triple negative has very high abundant expression of the glycosylated PD-L1. So we are wondering if we can generate antibody that can specifically target this bold structure of the PD-L1, we may generate more therapeutic impact. So in this regard, what we did is we inoculate glycosylated PD-L1 as an antigen into the mouse, and then we create 3,000 hybridoma clone. So then next, we use a flow cytometry to screen those 3,000 clones, try to look for the antibody that can specifically recognize glycosylated PD-L1, but not non-glycosylated PD-L1. And then we use the Intusight machine to help us to further narrow down which antibody has the ability to neutralize PD-L1 and the PD-1 interaction. We finally narrow down 2 monoclonal antibodies that has the ability. So this is a slide showing you that STM10004 and the STM108 antibody. These 2 antibodies has a very profound effect in inhibiting PD-L1 and the PD-1 interaction. So next, we would like to know where does these 2 antibodies bind to the PD-L1 glycan structure. So in this case, we create 3 NXT motif mutation, and we keep one, in this case is N35 as a wild type. And we keep individual glycan cell, glycan structure as a wild type, and then we did a simple Western blot. We found out that STM108 antibody can specifically recognize N192 and N200. And based on our earlier mass spectrum analysis, we already know that there is a very unique glycan structure appear on this N192 and N200, which are the two sites that can be recognized by our STM108 antibody. Because earlier studies show that glycosylation of the HER2 has the ability to cross-link the membrane-bound HER2 and therefore induce clathrin-dependent HER2 internalization. So this paper helps us to hypothesize that whether this our antibody, anti-glycosylated PD-L1 antibody, also have the ability to cross-link a membrane-bound PD-L1 and therefore induce this PD-L1 internalization and goes to the lysosome and further get degraded in the cells. So to do this, we incorporate with SM Bioscience technology and also with a pH rodel system. To do this, we fluorescence label our STM108 antibody with a fluorescence dye. And once this, so the theory is that when the cells start to uptake this antibody, the pH value will start from the membrane, which is pH 7, drop to the pH 4.5 inside the lysosome. And this acidic condition will help the linker region of the antibody to get cleaved. Once this linker region get cleaved, it will illuminate for red fluorescence in the microscope. So in this case, we are able to use a nucleoside lifetime imaging system to measure the fluorescence, red fluorescence, and also in the real time, we can take the picture how and where the red fluorescence appear. So in this case, we can see in 12 hours period of time, when we add the STM108 antibody into the BT509 stable clone, we can see very nice red fluorescence induced, suggest that STM108 antibody can specifically internalize into the cancer cell. And once it internalized, we can see we did a Western blot, and this was done in the after 5 days of treatment. We can see that significantly amount of the PD-L1 is reduced, suggests that this antibody has the ability to internalize PD-L1 and further degrade the PD-L1. So why this internalization and the degradation is so important? Because recently, there is a antibody drug conjugation technology that is available. So we are thinking whether we can take this advantage to conjugate cytotoxicity drug into our antibody. So in this case, we choose the NNAE as our warhead, and we conjugate. In our conjugation, on average, there are 4 chemo drugs being conjugated on our antibody. So we test the efficacy of this antibody in the vitro and also in vivo. We found out that only the cell has PD-L1 expression can specifically be treated by this antibody. This is in vivo, and here, when the mice has the PD-L1 expression, their tumor growth after treatment with our ADC is dramatically reduced. So this is the 41 human PD-L1 stable clone. They do not grow after we treat with our glycosylated ADC antibody. So this result is very exciting. So we also look at the tumor microenvironment to see that using the fluorescence IHC staining to detect the activated case page 3 activity. So in this case, in the red color, indicate the case page 3 activity. When the mice treated with glycosylated NNAE antibody, we can create a geometrical case page 3 activation in the tumor microarea. We also did a cytope analysis. We found that mice treated with the glycosylated NNAE antibody has dramatically tumor cell reduction, but slightly the immune cell get infected. So this result suggests that this antibody can selectively target the cancer cell, but not the immune cell, because as I mentioned earlier, that glycosylated or PD-L1 is highly abundant in the triple-negative breast cancer cells. So lastly, I would like to use this model to summarize this part of our study, which is that EGFR is the major signaling that can induce some sort of glycotransferase induction. And this glycotransferase will further glycosylate the PD-L1, and then this PD-L1 goes to the cell membrane. If we can create an antibody that specifically recognize glycosylated PD-L1, we'll have ability to induce this PD-L1 protein, and also once this endocytosis event happens, it will go to the lysosome and then get degraded. Once the antibody and the PD-L1 get degraded, the cancer cell cytotoxicity drug will be released, and then to kill the PD-L1 high expression cell, and also create a bystander effect, which will eradicate the cancer cell that do not express PD-L1. So this will create a profound therapeutic effect in reducing, eradicating triple-negative breast cancer. So the conclusion, what we found throughout this study, is that glycosylation is a very complex structure, and it's very difficult to assess in the regular basic research lab. So in this regard, glycosylation is often being neglected. So based on our signaling analysis, we can design a strategy and then try to isolate monoclonal antibody that can specifically recognize glycosylated PD-L1. And we first found out that this PD-L1 can cross-link membrane-bound PD-L1. So PD-L1 antibody can cross-link PD-L1 molecule and therefore induce the endocytosis of this protein. And through this endocytosis, we can let the antibody to carry a cytotoxicity drug and then induce a profound therapeutic impact to triple-negative breast cancer. So lastly, I would like to spend some time to acknowledge my mentor, Dr. Mien-Chie Hung at MD Anderson, and he is the world leading expert in study breast cancer for the past 20 years also. This project is collaborate with my colleague, Dr. Song-Wei Lin. We are contribute to this project equally. I would like to take this opportunity to thank ST-Cube Pharmaceutical. They have been provided a very powerful machine, and also they have a research team to help us sort out the methodology we are trying to develop in the lab. And lastly, I would like to thank Mabel, Savi, and Judy for their technical support. Without them, we will have this webinar event will not be possible. And I would like to thank you for your attention. Thank you.
Thank you, Dr. Lee, for your presentation. Before we begin the question and answer session, I want to advise that a series of poll questions will appear on your screen during the Q&A. You may select your answers, then close the poll questions by clicking on the X in the right corner. And a quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Lee will answer as many questions as time permits. And the first question is, can glycosylation of PD-L1 be a biomarker to predict immune therapy response?
Yes. That's a very good question. We do have such hypothesis, but we do not have any data to conclude that yet. So it is known that patients with more PD-L1 or interferon gamma expression in their tumor tissue, their response to immunotherapy is better. And most recently, actually, scientists at Johns Hopkins University, they also show that mismatch repair deficiency can accurately predict the anti-PD-1 treatment. So in that phase 2 clinical trials of study, they found out they conducted in the 41 colorectal cancer patients. They found out that mismatch repair deficiency can predict the higher the deficiency of the patient, they have around 40% of the response rate to anti-PD-1. So this is encouraging to us. So to answer your question directly, that this is certainly of interest to test whether our anti-glycosylated PD-L1 can be a non-invasive prediction marker or serial marker to predict future immunotherapy response. Yeah, thank you.
Why do you think glycosylation is so important to human oncology, or to human, excuse me, let me repeat that. Why do you think glycosylation is so important to immune oncology?
Yes, so as I mentioned earlier, that glycosylation has been neglected due to the lack of the technology. So that's also one of our primary interests. We want to develop, we want to explore this study through our finding on the PD-L1. We are expecting more and more immune receptor ligand interaction also require glycosylation. And also genetic mutation, neoantigen presentation, neoepitope presentation, T-cell activation, interferon gamma secretion. Those are the critical steps that immuno-oncologists are trying to understand right now. So glycosylation is known to involve in several processes of above mentioned process. So we believe that glycosylation has been largely neglected in the past, and we are committed to the field of this research, and we would like to invite whoever interested in this field also join us to answer important biological questions and to help patients in the bedside. Thank you.
Given the importance of interferon gamma signaling pathway in PD-L1 expression and PD-1 blockade, do you know if interferon gamma also induces PD-L1 stabilization similar to that of EGF?
Yes. This is a common question we get from our earlier publication. So we do compare both EGF and the interferon gamma induce the PD-L1 expression at either endogenous level or at the ectopic expression level. While both EGF and the interferon gamma can induce significant amount of endogenous PD-L1 expression in the triple-negative breast cancer cell per se, the protein stabilization was only found in the EGF stimulator cells. So together, this result kind of gives us a hint that there are two distinct mechanisms. One, in the EGF case, it only induces protein stabilization, whereas in the case of interferon gamma, they are inducing through the translational regulation. But what caused the difference is still not very clear yet. So we also have tremendous interest in understanding the underlying switch mechanism of these two signaling differences. Thank you.
Human PD-L1 in APC may play a major role in PD-L1 and PD-1 mediated immunosuppression. Do you know if PD-L1 glycosylation is also observed in APC?
As a matter of fact, we did not try to conclude that the suppression of the T cell activity is only through tumor PD-L1. There's a lot of evidence to show that those PD-L1 also expressed in other immune cells, such as macrophage or dendritic cell. They can also exert the T cell inhibition ability. But one thing we'd like to point out is that in our system, as I show you in the earlier slide, that we only focus on the cancer cell. So our system is not to address the way how important or whether it's not important of other immune cells. We are focused on the cancer cell. So right now we cannot exclude, and we do not try to exclude that PD-L1 in other type of immune cell also involved in such a case. And we believe, yes, EGF may also induce immune cell PD-L1 to suppress T cell through maybe similar mechanism or in distinct mechanism, which we do not know. So to answer your question, sure, is that it's our system, it's fundamental system, is not allowing us to address APC's question, and we are still open for that area. Thank you.
Do you know if glycosylation also happened on other immune checkpoint receptors?
Yes, that's important question. So as I mentioned earlier, that as many as 1/3 of human protein are all exist as a some sort of glycoprotein, either N-link, O-link, or C-link glycosylation. So through this study, yes, we focus on the PD-L1, but the message we would like to expand to deliver to people is that there are a lot more glycosylation happen on the immune cell receptor we should pay attention to. And we, as a matter of fact, we know that PD-1 also has the glycosylation, and we believe those glycan structures will, on the cell surface, will intertwine to each other, and then cause their interaction become very tight. So if we can develop a way, either use inhibitor, such as I just mentioned, maybe TKI, if we can block this glycan synthesis, is also another new way to help us to achieve reducing this PD-L1 and the PD-1 interaction. And also for your information, we do have study evidence, which the manuscript is in preparation right now, that we test another 22 set of receptor ligand interaction, and we do found one subtype of immune receptor, their glycosylation is important for their interaction, whereas another subtype, their interaction does not require for the glycosylation. Thank you.
Is glycosylation of PD-L1 the major signal in triple-negative breast cancer?
Is a major signal. We actually test them in many type of disease, melanoma and colon cancer. This glycosylation is also happen in many type of cancer of PD-L1. So, but in the triple negative case is that you can see a very dramatic difference from non-triple negative and the triple negative. But for other disease, some cell we can see the glycosylation, some cell we do not, but we cannot characterize them. We still, in a way, we still in a process of characterizing what kind of subtype of cell that has the glycosylation. Thank you.
Are there any other glycosylation targets in triple-negative breast cancer?
Okay, you are answering the, okay. I think that's a good question. So as I mentioned, we can either use a glycotransferase inhibitor or sugar analog or right now we are working on the ER stress inducer. We are thinking a much bigger picture that there will be more and more inhibitor that can be developed using this type of strategy we just developed in the lab. So we are still in a process of looking for different ways to try to see what can help us to inhibit this glycosylation and thereby enhance the T cell activity. Thank you.
What kind of antibody is used for detection of glycosylation?
This is a collaboration project with the ST-Cube Pharmaceutical. So we use, I above mentioned the method to identify the first anti-glycosylated antibody. So it's under proprietary protection of the antibody. And if you are interested in using this antibody, we will encourage you to communicate with ST-Cube Pharmaceutical to see if they are willing to share this antibody. But for your information, to detect the glycosylation, normal antibody, some of the antibody can recognize the glycosylation. The one we use in our paper mostly is the cell signaling antibody. So if you are interested in how we identify the glycosylation form, as the data I show you, you can check our Nature Communication 2016 and also our Cancer Cell 2016 paper. And we are happy to share our methodology or reagent or anything. Just for the data I present earlier in the slide, the STM108 antibody is a collaboration project with the ST-Cube. So for that antibody, we cannot share. Thank you.
What kind of antibody is used for detection of glycosylation? I'm sorry, did I just ask you that one? Here's the next one. Did you look at glycosylation of PD-L1 in macrophages?
Yes. This is a common question we get from the reviewer. We do have that data in the but somehow the PD-L1 expression level on the macrophage is not a lot. So if we induce the PD-L1 expression in the macrophage, we can see that PD-L1 also exists as a glycosylation form. Thank you.
TNF-alpha stabilizes PD-L1 through P65 transcription of CSN5. Does TNF-alpha also modulate glycosylation of PD-L1?
That's a good question. So right now we don't know. This is if you read our paper, the TNF-alpha stabilizes PD-L1 through post-translational modification on the ubiquitination. So we do not have strong evidence to link glycosylation and the stabilization. In that paper, we haven't investigated whether TNF-alpha also affects the glycosylation status of the PD-L1. So we have two papers, and those two papers we haven't linked together. But that's definitely a good question. Maybe in the future we are, if we have time, we'll also investigate that possibility. Thank you.
Is your anti-GPDL1 antibody suitable for CART1 cell therapy?
So recently, FDA has approved Novartis CAR-T therapy for ALL treatment. As you know, people in the field must have known that. So the result is very exciting that the response rate could reach 85% just in one shot of the treatment, which is a very promising result. And however, the price of the treatment right now is around $500,000 for one shot of the treatment. So right now we do not have. This is as I mentioned, this only happen in the leukemia, which is a liquid cancer. So people are trying to develop the CAR-T therapy in the solid tumor. So there is no CAR-T therapy in the solid tumor currently available. So this is definitely a good direction. And since we have the anti-glycosylated PD-L1 antibody, And we are definitely of interest to know whether we can develop a CAR-T therapy in the future to target triple-negative breast cancer, which is a type of solid tumor that is difficult to treat. And we are working on that. We are in the early stage of developing this direction. Thank you.
The next attendee has 3 questions, so I'll ask them one at a time. They say, hi, really great job. And then, do you think that your GPDL1 monoclonal antibody works better in TNB? Sorry. Hang on one second. My screen just jumped. Do you think that your GPDL1 monoclonal antibody works better in triple-negative breast cancers because these cells express high levels of PD-L1?
Yes, certainly. Yes, that's the reason. Thank you. As I showed earlier, that triple-negative has a very distinct PD-L1. They are all highly expressed glycosylation form. So that's our hypothesis. That's the reason we want to generate glycosylation-specific anti-PD-L1 antibody. And we believe this could, we haven't do, side-by-side compare with non-glycosylated PD-L1 in terms of therapy. We believe this will provide another different application to current anti-PD-L1 therapy. Thank you.
And the next part is, could the high level of PD-L1 in cancer cells be due to the intrinsic genomic instability of these cells?
Yes. So as I mentioned, genomic instability causes a lot of mutation. This is difficult to approach because glycosylation is so complex. So we do feel that's the strong link, because genomic stability causes a glycan structural change and therefore affect PD-1, PD-L1 interaction. So that's also the message we would like to develop is that the quality of the PD-L1 may be more important than the quantity of the PD-L1. So therefore, as a basic scientist, it's important to really understand what the glycan structure and how those glycan structure happen that way through genomic stability and cause certain glycosylation moiety happen on the PD-L1, and that contribute to immunosuppression. That's a very difficult question, but we would like to further pursue. Thank you.
Okay, so the last one from this person is, what do you think about a combination of radiotherapy and PD-L1 antibody to kill cancer cells?
I think those has been approved. Some of the literature I remember already prove some of the concept that the radiotherapy will create more neoantigen genomic instability and then have more neoantigen, and therefore cancer cell has more immune response to it will attract more T cell into the tumor microarea. So with that, we predict that the PD-L1 response will be much higher in the presence of radiotherapy. I think, in fact, Dr. Andy Mien already published a paper in the Cell, I think 2016, also talk about, he has multiple papers, all deal with the radiotherapy plus anti-CTLA-4 immunotherapy. I think these are very similar direction. So if you are interested on that, I suggest you to maybe read more of the Dr. Andy Mien's paper. And if you still have questions, cannot find the paper, feel free to contact us. We are happy to share the knowledge, and thank you for the question.
I would like to once again thank Dr. Lee for his presentation. Do you have any final comments?
Yes, thank you for having me to have this opportunity to present some of our recent work. And we are very excited that this is a simple, straightforward finding, and we would like to develop, and we would like to collaborate. So whoever in the audience, if you know the concept, if you have questions, if you have any suggestions, please contact us, and we are willing and happy to share reagent or discuss the science, or in the possible future, if we can create collaboration or brainstorm to each other and benefit each other's project, that would be tremendous. So again, thank you, and I really appreciate this event.
Can I throw one more question at you? We're going to close out. How about glycosylated EGFR's effect? Let's see. How about glycosylated EGFR effect, PD-L1 expression in triple-negative breast cancer? So let's see. To phrase it, how does glycosylated EGFR affect PD-L1 expression in triple-negative breast cancer? How's that?
So the question is, how does glycosylated EGFR? Yes, EGFR also get glycosylated, and but it's not so far we haven't gone to that stage to study the EGFR glycosylation. We know when we treat with tunicamycin, the expression of EGFR will reduce, but we have not looked into to compare, and which is a good question. I mean, that's very beneficial to our research. That is to compare the response of the glycosylated form of EGFR and non-glycosylated form of EGFR in the presence of EGF. I think that's a really good question, and right now we don't know because that's involved in ligand and the receptor interaction, whether the glycan structure of the EGFR have anything to do with the ligand association, which is we have to test. So to answer this question quickly, as I mentioned earlier, we do test the different receptor and the ligand pair in 22 cases. So some glycosylation affect the protein-protein interaction, and in some cases, glycosylation is not involved. So for EGF and the EGFR, whether the glycan structure of the EGFR affect EGF binding, we have no idea. And thank you.
Thank you again, Dr. Lee, and I'd also like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, SN Bioscience, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through March 7th, 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.